Hello everyone, welcome back to my channel Suggesti Tutorials. Today in this video session, we are going to discuss about the structure and functions of different cell organelles which are present in the eukaryotic cell. At first, we are going to start with the endoplasmic reticulum. Endoplasmic reticulum. The endoplasmic reticulum is also called as ER. ER means endoplasmic reticulum. And the endoplasmic reticulum are tubular-like structures. The appearance of endoplasmic reticulum are tube-like structures. And where these endoplasmic reticulum are present in the eukaryotic cell? Let us assume this as a nucleus. And the membrane which surrounding the nucleus is called as nuclear envelope. This is the nuclear envelope. And the nuclear envelope is surrounded by tubular-like structures. What are those tubular-like structures? So this is the nuclear envelope, and these are the tubular-like structures which are getting surrounding the nuclear envelope. What are these tubular-like structures which are getting surrounding to the nuclear envelope? These are called as endoplasmic reticulum. As we have observed now that half portion of the endoplasmic reticulum is attached to the nuclear envelope and remaining half portion of the endoplasmic reticulum is observed freely floating in the cytoplasm. So half the portion is attached to the nuclear envelope and half the portion is present in the cytoplasm. This is the cytoplasm. So, coming to the location of the endoplasmic reticulum, the endoplasmic reticulum is observed attached to the nuclear envelope. Attached to the nuclear envelope. The endoplasmic reticulum is divided into two regions. What are those two regions? Luminal region and extraluminal region. What is the luminal region? The area which is present inside the endoplasmic reticulum. The interior region of the endoplasmic reticulum is called as luminal region. Inside the ER. So, the interior portion of the endoplasmic reticulum is called as luminal region, and the extraluminal region is the region which is present in the cytoplasm. So, the endoplasmic reticulum which is observed in the cytoplasm is called as extraluminal space in the cytoplasm. The endoplasmic reticulum which is observed in the cytoplasm is called as extraluminal space region, and the region which is observed inside the endoplasmic reticulum is called as luminal space. And coming to the types of endoplasmic reticulum. There are two types of endoplasmic reticulum. Already we have discussed about the types of endoplasmic reticulum in the earlier class. What are those types? SER and RER. What is SER and RER? SER means smooth endoplasmic reticulum and RER means rough endoplasmic reticulum. What is smooth endoplasmic reticulum? If the surface of the endoplasmic reticulum does not contain ribosomes, then it is called as smooth endoplasmic reticulum. So, the surface of the endoplasmic reticulum does not contain ribosomes. No ribosomes in the smooth endoplasmic reticulum. If the surface of the endoplasmic reticulum contains small granular-like structures, the surface of the endoplasmic, some of the endoplasmic reticulum surface is surrounded by small granular-like structures. what are those granular-like structures are called as? Those granular-like structures which are present on the surface of the endoplasmic reticulum are called as ribosomes. If the surface of the endoplasmic reticulum are covered with the ribosomes, then those endoplasmic reticulum are named as rough endoplasmic reticulum. So here ribosomes are present. Where the ribosomes are present? The ribosomes are present on the surface of the endoplasmic reticulum. Then those type of endoplasmic reticulum are called as RER, means rough endoplasmic reticulum. So this is the complete structure of the endoplasmic reticulum. Then coming to the function of the endoplasmic reticulum. What is the function of the endoplasmic reticulum? Here we have already discussed that endoplasmic reticulum is divided into smooth endoplasmic reticulum as well as the rough endoplasmic reticulum. Coming to the smooth endoplasmic reticulum. Here in the smooth endoplasmic reticulum, lipids formation takes place. So formation of lipids takes place in the smooth endoplasmic reticulum. So what are the lipids that are formed in the smooth endoplasmic reticulum? Steroid hormones are also synthesized in the smooth endoplasmic reticulum. And coming to the RER, what happens in the RER? What is the function of the rough endoplasmic reticulum? In the rough endoplasmic reticulum, protein synthesis takes place. Protein synthesis takes place in the rough endoplasmic reticulum. Here in the smooth endoplasmic reticulum, lipids formation takes place, whereas in the rough endoplasmic reticulum, proteins formation takes place. The proteins and lipids which are formed in the endoplasmic reticulum are phospholipids and phosphoproteins. So this is about the complete structure and functional details of the cell organelle which is called as endoplasmic reticulum. And now we are going to discuss about the second cell organelle which is present inside the eukaryotic cell, that is the Golgi complex. The Golgi complex is the densely stained reticular structures. So the Golgi complex are the densely stained reticular structures. They observe like reticular like network like manner, and where this Golgi complex is located in the eukaryotic cell, the Golgi complex is observed near the nucleus. Where this Golgi complex is present? This Golgi complex is present near the nucleus, and the first who observed this Golgi complex in the eukaryotic cell was Camillo Golgi. Camillo Golgi. Camillo Golgi was the first to observe the densely stained reticular-like structures near the nucleus and named them as Golgi complex. Coming to the structure of the Golgi complex. So, the Golgi complex looks like parallel arranged reticular-like structure. So, see here, the cells are arranged parallelly. One after the other, the layers, reticular-like layers are arranged. And the flat stack-like structures which are observed in the Golgi complex are called as cisternae. These structures are called as cisternae. These are flat-like structures which are arranged parallel to each other. So, the flat-like structures which are arranged parallelly are called as cisternae. Are called as cisternae. Again, the Golgi complex region is divided into two phases. Means the regions of the Golgi complex is divided into two phases. What are those two phases? Cis phase and trans phase. What is the cis phase and trans phase? The cis phase is the entry point for the proteins and lipids phase, and the trans phase is the exit point of lipids and proteins. Why they are entering the cis phase and exiting from the trans phase? The proteins and lipids which are formed in the endoplasmic reticulum enters into the cis phase. So this region is called as cis phase, and the exit point is called as trans phase. So in the cis phase, the proteins and lipids which are formed in the endoplasmic reticulum enters into the Golgi complex through the cis phase. So what happens in the cis phase? The lipids and proteins which are formed in the endoplasmic reticulum gets enter into the Golgi complex through the cis phase. After entering through into the Golgi complex through the cis phase, they move into the trans phase. Here, the processing of lipids and proteins takes place in the trans phase. So the cis phase is called as entry point, and whereas the trans phase is called as exit point. From the cis phase, the proteins and lipids gets enters into the Golgi complex, and after entering into the Golgi complex, they move to the trans phase of the Golgi complex. Here, the lipids and proteins get processed. Processed means here the lipids and proteins forms into vesicle-like structures. Those vesicle-like structures gets removed out of the Golgi complex, and those vesicular-like lipids and proteins move to the other cell organelles of the cell. So this is about complete function of the cis phase and trans phase of Golgi complex. So again I am repeating, the Golgi complex contains two phases, namely cis phase and trans phase. What happens in the cis phase? The lipids and proteins which are formed in the endoplasmic reticulum gets enter into the cis phase of the Golgi complex. From the cis phase, the proteins and lipids enters into the trans phase. Here in the trans phase, the proteins and lipids gets processed. Processed means the proteins and lipids gets forms into vesicle-like structures, and those vesicle-like structures comes out of the Golgi complex through the trans phase, and they get moves to other cell organelles which are present in the eukaryotic cell. This is about the complete structure and function of the Golgi complex. And now we are going to discuss about the another cell organelle which is present in the eukaryotic cell, that is lysosomes. And now we are going to discuss about the another cell organelle which is present in the eukaryotic cell, that is lysosomes. The lysosomes are spherical or round vesicular-like structures. The lysosomes are vesicle or round spherical-like structures. Where these lysosomes are present? These lysosomes are present freely floating in the cytoplasm. These are observed freely floating in the cytoplasm. So, the lysosomes are the vesicle or round spherical-like structures, and these are observed freely floating in the cytoplasm. And how these vesicular-like structures are formed? How these lysosomes are formed? Already we have discussed that earlier that Golgi complex leads to the formation of processing of proteins and lipids. Those processed proteins and lipids gets formed into vesicle-like structure. Similar way, in the Golgi complex, not only lipids and proteins processing happens, but also hydrolytic enzymes are also processed in the Golgi complex. Hydrolytic enzymes. What are the hydrolytic enzymes? Lipases, proteases, carbohydrates. These are the different types of hydrolytic enzymes which are processed earlier in the Golgi complex. The processed proteins, lipids and carbohydrates forms into vesicle-like structures. Those vesicle-like structures which contains the hydrolytic enzymes are called as lysosomes. So, the vesicle-like structures which contains the hydrolytic enzymes are called as lysosomes. What are the hydrolytic enzymes which are present in the lysosomes? Lipases, proteases and also the carbohydrates. What is the function of the lysosomes? The main function of the lysosomes are they act as digestive system. The lysosomes act as digestive system. What is the digestive system function? The digestive system, what happens in our digestive system? The proteins, lipids and also the carbohydrates gets break down in the digestive system. In that similar way, the lysosomes also breaks the food particles into smaller particles. So, in the lysosomes, the digestion takes place. Means the breakdown of food particles takes place. And they also act as defense mechanism. What is the defense mechanism? If any bacteria or virus enters into the eukaryotic cell, then these lysosomes break those bacteria and virus into smaller pieces. So, the lysosomes also act as defense mechanism. So, if any bacteria or virus enters into the eukaryotic cell, then these lysosomes break those bacteria or viruses into smaller pieces. And the digestion which takes place in the lysosomes are of two types. Intercellular digestion and second is the intracellular digestion. What is the intercellular digestion? We have already discussed it that the eukaryotic cell contains a number of cell organelles. If any cell organelles gets damaged or becomes old, then those damaged cell organelles are broken down into small pieces and these lysosomes cleans the damaged cell organelles and remove those cell organelles from the eukaryotic cell. So intercellular digestion means the digestion which occurs within the cell. So the digestion which takes place inside the cell is called as intercellular digestion. What is the intercellular digestion? If any cell organelle gets damaged in the eukaryotic cell, the damaged cell organelle gets broken down by the lysosomes and those broken down pieces are removed from the eukaryotic cell by the lysosome. That is called as intercellular digestion. The digestion which occurs within the cell. And the intracellular digestion means the digestion which takes place out of the cell is called as intracellular digestion. Outside the cell. So what is the outside the cell? The eukaryotic cell takes some of the food for the energy required process. So if any bacterium or any dust particles enters into the eukaryotic cell from the outside, then the lysosomes breaks down those dust particles or the foreign particle into pieces. So This is called as intracellular digestion. The digestion which takes place outside the cell. If any foreign particle enters the body, the lysosomes helps for the breakdown process and removes those foreign particle from the eukaryotic cell. So this is about the complete structure and function of the lysosomes. And now we are going to discuss about the another cell organelle which is present in the eukaryotic cell, that is the vacuole. The vacuole are the membrane bound structure. Membrane bound structure means the structure of the vacuole is surrounded by membranes. Vacuole is the membrane bound structure. So this is the vacuole, and the layer which gives protection to the vacuole is called as tonoplast. This is called as tonoplast. This type of vacuole is present in the plant cell. So this is the vacuole which is present in the plant cell, and the outer layer which is surrounding the vacuole is called as tonoplast. And what is the function of the tonoplast? The tonoplast helps for the movement of ions and minerals into the vacuole. So what is the function of the tonoplast in the plant cell? Ions and minerals are moved inside the vacuoles through the layer called as tonoplast. And in the plants, the vacuole is very large. Very large. What are the components that are present inside the vacuole? The vacuole contains water, sap, and also other minerals are also present inside the vacuole. Sap means the fluid which contains sugars and salts, that is called as sap. So inside the vacuole there is presence of water and also sap, and other dead materials are also stored in the vacuole. So what are the components that are present inside the vacuole? Components of the vacuole: water, sap, and also other minerals and also waste materials which are not used by the eukaryotic cells get stored in the vacuole. We have already discussed that the plant vacuole contains the protective layer called as tonoplast. And what is the function of the tonoplast? Different types of ions and minerals get inside the vacuole through the help of the tonoplast. And coming to the vacuole which is present in the amoeba. The vacuole which is present in the amoeba is called as contractile vacuole. Contractile vacuole. What is a contractile vacuole? And the contractile vacuole regulates the osmoregulation process. Osmoregulation process. So what is osmoregulation process? If the vacuole contains excess of water, then those excess of water is removed through the help of this contractile vacuole. So this is the vacuole, and if the vacuole contains excess of water, then this excess of water is removed through the contractile vacuole in the amoeba. And coming to the protista. In the protista, the type of vacuole which is present in the protista is called as food vacuole. The type of vacuole which is present in the protista is called as food vacuole. What is the food vacuole? In the food vacuole, the food gets stored in the vacuole. So, if the protista wants to eat food any other time, then the food gets stored in the food vacuole. So, what is the function of the food vacuole in the protista? Stores food. Whereas a contractile vacuole, what is the function of the contractile vacuole in the amoeba? The contractile vacuole helps to remove the excess water which is present inside the vacuole. So, this is about the complete structure and function of the vacuole. Hope you all have a clear idea about the cell organelles that which we are discussed in this video. If you like this video, hit a like button, share to your friends and subscribe to such tutorials. Thank you.